on this computer. I hate technology.
That's okay.
Let me make sure I don't have any revealing tabs open. No, I probably should have done that before I started recording. So this is week 4. There's only 2 points we're going through today, which is pop health. It's study design, isn't it?
Yeah. Study design stuff, yeah.
And then we're doing some micro, which is pathogenesis, which is also, it's fairly low yield, so you don't have to stress too much. So yeah, without further ado, we'll get started.
So for study designs, I think it's like one of, okay, in general, pop health is actually fairly examinable, but study design is like the main one. So on the next slide, there are just a few key definitions. These are probably things you would kind of know by now, but just I put them up there just in case. So case is someone who either like has a disease, like we consider them to be like a case, or they're a person of interest for whatever reason, like related to your study. Control, back to your 9 science, is the thing you're comparing the case to, so they don't have a disease or they're not a person of interest. Exposure is like the factor or the thing that the people are exposed to that you think makes them get the disease. So for instance, if your disease is like liver failure or something, exposure could be like alcohol, because alcohol can cause like liver failure. Outcome is just the result of a study. So in epi, they usually talk about like exposures and outcomes, and then they do like an odds ratio with that. So those are just some definitions. On the next slide, you can see kind of all the study designs you sort of need to know. And it's good to know them in that category, I guess, on the left, but that pyramid on the right is actually what you kind of need to know more than the thing on the left. So pretty much what they can ask you about each study design is like what it is, like what it entails. And you guys have an assignment coming up soon where you have to like, you get given like a study, you have to like do like all these pop health questions on it, like what study design is, what's the exposure, what results do you have? So that's where like all this comes into handy. And they also sometimes give you a description of a study design on your exam, and you just have like a multi-choice or like an extended response, where they have like all the study designs listed out, and you have to say which one is which. On the right, that pyramid there is your level of evidence hierarchy. So what that means is that the further up you get, the better it is like as a science. So down below, you see like ideas, editorials, opinions. Obviously, someone's opinion isn't like a very good study at all. Then you have like case reports, case series, ecological studies. These are like all your observational descriptives. Then you go on to your analytical, which is your cross-sectional studies, your case control and your cohort. And then you go on to your experimental, which is your RCT. You don't talk too much about meta-analysis yet. It comes up a bit later this semester. And field trials and community trials, at least we didn't talk about it at all. So it wasn't relevant for us. Lovely, David. We'll just go through them now one by one. So yeah, ideas, editorial opinions, really bad. Obviously, are the lowest level of evidence, are very biased, and we kind of disregard them. Next one. Okay, so this is a little bit more relevant. For cases, we have kind of 2 studies you can do with them. You can do a case series or a case report. The only difference is the number of patients. Case series is you have multiple patients. Case report is you have 1 patient. And pretty much the whole idea of the case is that usually someone comes to the doctor, they have like a list of symptoms, it doesn't fit a known disease. The doctor pretty much just writes down a description of like all the symptoms and all like the things they think could be causing it. And it's like just sharing with the community, this is what I observed, but it's not an experiment in any way. He doesn't know exactly what's causing what's happening. But pretty much it's a good way to identify like new diseases or rare diseases, because to do an experiment, you generally need like a large number of people. And if you have like a rare disease, maybe only 1 person has it, so it's a bit harder. And it's good as like a starting point for like other studies. So some studies might look at these case reports and decide, oh, I could do a study on the relationship between exercise and I don't know, mental health, based on like your case reports. Lovely, next one. Ecological studies are when you're looking at a population. So I don't know about you guys, but when I was doing like psych in BC, we kind of learned a different definition of ecological studies. So throw that out the window. Ecological studies in med, at least, or in epi, is when you're just looking at an exposure and an outcome happening to like a group of population. So the idea is you're trying to link an exposure and you're trying to link an outcome with a large group of people. So I don't know, for instance, saying that all people in rural Victoria have hypertrophy or something. So like trying to link it to a large group. The reason it doesn't work is that it's not strictly like an experiment. It's more like an observation that you're seeing with a pattern of a large group of people. Different populations work in different ways. So even though your testing population doesn't work, and you have a lot of confounding variables and bias, which we'll talk about next week. The next one is cross-sectional. Here, it gets to be a little bit more like an experiment. It's not quite an experiment, but it's a little bit more relevant in that you pretty much grab a group of random people, and then you try to figure out, okay, which ones have like a disease, if you like studying a particular disease, like which one of these have cancer, let's say breast cancer, and which one of these were exposed to like, I don't know, some kind of carcinogen. And you're just taking a group of people at one time and trying to collect that data. So for instance, in below you see an example of depression and exercise. So you're taking a group of people and like, okay, 50 of them have depression. Of those who have depression, so they have the disease, maybe like 20 exercise, 30 didn't exercise. Of those who didn't have depression, maybe more exercise. So just trying to collect a group of people and try to figure out which one has an exposure, which one has a disease. And you can pretty much choose an exposure and a disease. The problem is here, we don't actually know what's the cause and what's an effect. So we think something's an exposure, but we don't actually know. So like with depression and exercise, does a lack of exercise contribute to depression, or does depression contribute to a lack of exercise? So it's a bit harder with a cross-sectional study. Lovely, next one. Oh, by the way, these blue diagrams you see kind of in the top right corner. The reason we have them there is that you'd have them in your lectures and also in your assignments, you have to like draw out a diagram. So these are like the standard diagrams for each of these. So a case control is when you select people that have developed a disease, so they're called cases, and people from the same population without the disease, and you ask them about their exposures. So you can see over there we have like time is going forward and direction of inquiry is going backwards. The direction of inquiry going backwards means that these people have already developed the illness or the disease, and then you try and figure out what caused it. So it's like you always have to go backwards in their lives, because to a time when they didn't have the disease, then what caused them to have the disease. The good thing about the case control is that it's relatively quick, and it's relatively easy to find people with a disease and without the disease. So if you're looking for like, I don't know, cancer, you can find people with cancer, people without cancer from the same population. So people age 60 to 70, for instance. So in that sense, it's like much more easy to do. The next one we'll go on to, which is like a cohort, which is a bit harder. The outcome here is reported as an odds ratio, and you'll go through that in your biostats lecture in a few weeks. Pretty much, it just means if you were exposed, how much more times are you likely to get the disease? We'll go through that later on. Cons is that recall bias is huge. So something like, like cancer. If let's say we're thinking, oh, maybe alcohol is a contributing factor. Asking people, how much did you drink 3 years ago before you're diagnosed with cancer? For the previous 10 years, how much did you drink per week? Like, it's a bit hard to get people to recall that far back. And usually their recall is a bit skewed given the disease. So recall bias is definitely huge in case control. Next, we have a cohort study. There are two cohort studies, types of cohort studies. The first is prospective, which means you're going forward. And the second one is retrospective, which means you're going backwards. In prospective, you choose a group of people. So you choose, I don't know, 20 18-year-olds. And then maybe half of them go work in a mine and half of them don't. And then later on, maybe in like 20 years, you see how many have developed lung cancer because of like that work in the mine. Now you can obviously see a problem with that. Firstly, there's no guarantee that they'll develop the disease. So the people you choose at the start might not even get the disease later on. Secondly, time takes forever because you get to them when they're healthy and you can't predict exactly when the disease will occur. And the second thing as well is that you have to let them be exposed to the exposure themselves. So you can't like demand people to go work in a mine. Like they have to like voluntarily choose to go to work in a mine. So you have to, it takes a while to find like a good group of people where you know half of them will go do something and the other half won't do something. Because it's hard to control those variables. And pretty much you can only use this for like common diseases, because rare diseases you can't really figure out who's going to develop it or at what point. So before, for the other one, it was reported as an odds ratio. This one, the outcome is reported as a relative risk. So don't worry too much about these words. I just put them there mainly for like revision later on when you talk about odds ratio and relative risk. I know you guys haven't studied it yet. It's going to be in like a biostats tutorial in a few weeks. Next one, retrospective. So retrospective is just when, so in prospective, oh, okay, lovely. David, maybe in the chat, can you write the difference between OR, odds ratio, and a relative risk? I have a student ask a question, and then I'll talk about it later on.
Sure, I just can't change the screen when we do it. So
oh, no, totally, that's okay. Okay, I'll do it when I finish. But yeah, thank you for that question. I'll answer it later on. Retrospective is when you're asking about things that have happened in the past. Cohort retrospective study is quite rare. Like you usually do a prospective cohort, prospective cohort study instead of a retrospective one. A retrospective study requires records of past exposure, and that can be traced to their current health. So for instance, if I grab a group of healthy people, but I know half of them were exposed to like a nuclear bomb or something, and I know they're exposed to nuclear radiation, that's retrospective because I already know the exposure happened, and then like I wait for them to develop the disease. That's kind of where it happens. Whereas in prospective, like it's before they've been exposed to anything. So here, recall bias as well is massive because people can record that they've been exposed to something, that they haven't been exposed to something incorrectly. And it's sometimes used in the military, a fun fact. All right, next one. RCT, this is your gold standard. If you ever asked, which one in ideal conditions would you like to do? RCT is the one you always choose, hands down. But the only like problem with it is like the ethics. So RCT is like your standard science experiment in high school. You have a population, you have half of them exposed, half of them not exposed. But unlike in a cohort, you have to like purposely expose them, if that makes sense. Because in an exam, in a controlled experiment, like you're the one that decides who's exposed, who's not exposed. And then you see who develops the disease. Now, obviously, there are like problems with this. So let's say you want to test how alcohol affects babies being born. You grab a group of like pregnant women, that's your population. You can't then like randomly allocate half of them to drink alcohol, because that's like bad. It's like really unethical, especially if you know that that could cause harm. So something like that, you'd probably do a retrospective cohort study, or you'll follow a group of pregnant women. And in retrospective, you would have asked, oh no, did you drink alcohol? And those who did, well, there you're exposed then. If you did a prospective study, then you would grab a group of women, see who drinks alcohol, and then see what happens. But in a controlled trial, you're like actually allocating them to these groups. But it's the best science like available out there. It's the most like accurate one to use, the highest level of evidence. And for experiments that are like have a lower harm, I guess, then this is a good version. Or like, for instance, something that like might be fair here is taking like different vaccines, like exposed to the Pfizer vaccine and then not exposed to Pfizer, but exposed to like a different one, and seeing like the difference between vaccines. Like that could be okay because that's less unethical than like not giving anyone the vaccine. All right, next slide. Meta-analysis and systematic review. So everything we've discussed so far, super quickly, so sorry for going so fast, is like a single kind of case or study or an experiment. Meta-analysis or systematic review is a combination of like different experiments and you're doing something to them. So meta-analysis is all about the numbers. Pretty much you take all the results from all these experiments. So maybe you have like 10 experiments on, I don't know, how sleep affects cognition, like your reaction time or something. And then after a meta-analysis, you get a number. You're like, okay, if you don't sleep for 10 hours, your reaction time is reduced by 0.5 seconds. Like you get a number at the end. That's what a meta-analysis is. A systematic review is more like a literature review. You read all the different trials and you kind of just like synthesize the answers. Systematic review doesn't necessarily give you like a number at the end. It more just gives you like a broad conclusion. And it's particularly good for stuff like qualitative stuff, like stuff that has like words and descriptions rather than like actual numbers. So like how much more happy are people who have kids than people who don't have kids. So you might get like a conclusion like in general, people report being more happy with no kids. So you get more like a wordy answer, whereas meta-analysis you actually get a number at the end. And in some systematic reviews, they include a meta-analysis. So as part of the results, they have a meta-analysis in there. And I think that's it. Yeah, that's it. All right, I'll answer some of the questions in the chat.
Yeah, what we can also do is we can show them with the canvas. I just don't know how to put a canvas up.
Canvas?
Like when you can draw on this, so we can draw to show
like a
them.
whiteboard?
Yeah.
Oh.
Do you know how to do that?
Yeah, just stop sharing and share screen whiteboard. But what should we draw?
I'll draw the.
Go, draw it.
To my very limited understanding, the difference between a risk ratio, relative risk, and odds ratio.
Odds ratio.
Interrupt me if I'm wrong.
Yeah, totally. But I feel like you got this.
This is an intervention. This is control. Let's imagine that the intervention is smoking cigarettes. The control is not smoking cigarettes. The outcome is cancer. Maybe what I'll do is I'll I know. I'm just going to type it in text so it's legible.
Yeah, fair enough.
Cancer, not cancer. A is a number of people who had the intervention who developed cancer. B is the number of people who smoked who didn't. C is the number of people who didn't but still developed cancer. D is the number who didn't and didn't develop cancer. If you want a relative risk would be A on A plus B. In other words, it's the amount of people with the intervention who develop cancer divided by the total amount of people who experience cigarettes. That's like a probability. You divide that by the amount of people who in the control group who develop cancer divided by the number of people in the control group. You then take this number and you put it over this number. If it's
can't see a
equal to
mouse,
1,
by the way.
sorry?
Oh, we just can't see a mouse. Just so.
Right. Thank you for that. So let me just annotate. So draw Oh, it didn't even come up. Sorry, my
Oh,
fault.
now it's up. Now it's up.
Awesome. So this A is the amount of people in the intervention who develop cancer out of the total amount of people in the intervention. So it's the relative risk of developing cancer from smoking. C on C plus D is the relative risk of developing cancer without smoking. You compare the top one with the bottom one. If it's equal to 1, the relative risk is 1. There is no risk. The risk is the same. If it's greater than 1, this ratio, then there's a greater risk. If it's less than 1, it's actually protective. Odds ratio is a little bit different to my understanding. Odds ratio would be A divided by B.
Yeah.
That was destroyed. A divided by B, which is the people in the intervention who got cancer compared the odds compared to the people in the intervention who did not get cancer, divided by the control who got cancer divided by the people who didn't get cancer. This is saying the odds of the people who got cancer, what is the chance that they smoked? If it's 1, there's a high, if it's greater than 1, smoking meant there was a high, if they had cancer, there was a higher chance they smoked. If it's less than 1, it's protected. This is really complicated and you have a whole tutorial on it. So, Kat, do you have anything to add? I don't know how helpful that was. I hope it was.
No, David, that was really, really good. As David said, it takes a while to wrap your head around it a bit. Drawing out like ABC really helped me personally. And then like trying to explain where you're at. They give you the tables. So you just have to like sort of calculate it from there. And they usually give you like a study design, so you sort of know based on the study design which one you're supposed to calculate as well. The relative risk or the odds ratio. But so you explain it really well.
So usually they'll say case control and then you'll know the answers, odds ratio. Or they'll say cohort study and you'll know the answers relative risk. So you'll rarely have to figure it out from first principles. You just learn it.
Yeah.
Awesome. So I hope that was helpful. If you have questions, hit us up in that forum. I haven't actually looked at the forum yet. I should do that in our Google form. I'll look at that after this. So I made a mistake last week and I gave a lecture on pathogenesis 3 instead of 2. So now I've come back to 2. If you feel like you know it already, sorry about that. Give it a try. If you're bored, you can do what you like, but we'll go over 2 just for completeness because I don't want people to just rely on this and then not get the full information. There's 2 components of the immune system in the innate immune system that are quite important in regards to factors, which are like little proteins. There are cellular factors, which involve macrophages, polymorphs or neutrophils, and natural killer cells. Neutrophils, basophils, and eosinophils are polymorphs or granular cells. You also have natural killer cells, which are innate systems that go around and kill cells. Phagocytosis is a process of eating and killing of a pathogen. A soluble factor is little proteins or cytokines that are secreted. They enable the acute inflammatory system, complement cascade, and clotting system. I said factors or proteins. I mean soluble factors or proteins. These are cellular factors. These are cellular products like macrophages. That was a bit everywhere, but it's pretty simple. So hopefully it got across. This is important, probably the most important thing of this lecture. And this is the process in which phagocytosis occurs. So a microbe will adhere to a phagocyte. Often there's something called adhesin on this microbe that's going to interact with glycoproteins on the phagocyte, and that's going to pull it in. And there's going to be a process of endocytosis through the use of pseudopods. Pseudopods can also occur through the response of cytokines that can reach out to cytokines secreted by the bacteria. They're going to endocytose the bacteria, and the bacteria will be in a phagocytic vesicle called the phagosome. Sometimes all that's important is the antigen, which is that protein, which is a unique identity of the pathogen. An antigen can just be a flagellum or some identity, some protein identity of it. So you need to know the word phagosome is when the phagocyte is in a vesicle in the when the antigen is in a vesicle in the phagocyte. Hopefully that makes sense. That phagosome is going to combine with a lysosome to form a phagolysosome. And in a phagolysosome, all the lytic enzymes from this lysosome will degrade this pathogen. They fuse and it's killed by lysosomal enzymes, and then these are just excreted and removed by exocytosis. You need to know the words phagosome That's when it's by itself. Phagolysosome is when it combines with the lysosome. Probably the most high yield from this lecture. How does it actually kill? How does the macrophage actually phagocytose and kill pathogens? It has a low pH, it has lysosomes and hydrolases, which break down proteins mainly. And they're also very protective. And you have reactive oxygen compounds, which are highly toxic. The main ones to know are hydroxyl radicals and hydrogen peroxide, and they can destroy pathogens. Less high yield ones to know are reactive nitrogen compounds, antimicrobial peptides like defensins, which can destroy bacteria, and iron protein competitors because iron is necessary for the life of the bacteria. So no, low pH, enzymes, reactive oxygen. Opsonization is just the process of coating and marking a pathogen, and it's often done to increase the ease at which phagocytosis occurs, often carried out by complement of antibodies. It facilitates uptake, which will then lead to promotion of degradation. You can see here that the type of opsonization increases the degree of binding, which ultimately makes the process more effective. So these are just non-specific receptors. It's like not that strong binding. It's a less efficient process. Antibodies are more efficient than that. Antibodies and co-receptors are exceptionally efficient, and especially when there's complement as well. Because complement, as you would remember, you have the membrane attack complement and you have the opsonization complement. So usually the A's, so if you C3A, C5A, inflammatory, C3B, opsonization, C5 to C9, membrane attack complex, which punctures pore into the bacteria. These are some evasive mechanisms of phagocytosis. There's a lot: toxin, opsonization, prevention. You don't need to know all of these. All you need to know is that what exists are evasion mechanisms. Some bacteria can evade phagocytosis, and we'll talk about the methods they do this. One is through capsules. Some bacteria have a polysaccharide coat. It's negatively charged. It can mimic host molecules by adopting hyaluronic acid and salicylic acid. And what's scary is as soon as I made this presentation, I got an ad on YouTube for hyaluronic acid. So they're clearly watching me. The capsule can also evade recognition just physically, even. If you imagine there's a bacteria and there's a large sugary coating around it, the immune cells can't reach it and interact with its antigens. It's useful because it stops phagocytic uptake, stops complement, and stops antibody. It also mimics the host and evades the human immune response. They're intracellular pathogens, which actually do get uptaken into phagocytic cells, and they can also, as a side, invade other cells. Some are facultative. They live inside and outside cells. So Listeria and Salmonella can both live inside and outside a cell, whereas others always have to exist inside a cell. These are hard to treat and are protected means of replication and invention. Listeria, a bit more high yield. Listeria is a kind of a very serious food poisoning that's especially dangerous to pregnant women as it can damage the unborn fetus. So this is one method of pathogenesis, is through an intracellular escape, in which it's hard for the immune system to interact with them. Furthermore, pathogens can escape into the cytosol when phagocytosed. If you remember the phagosome, they escape the phagosome and they reside in the cytosol. They avoid toxicity of the phagolysosome, which is when the lysosome combines with the phagosome, and they can then move within the cell and spread to neighboring cells through actin polymerization. And this way they evade soluble factors and antibodies and complement. This is just a picture showing you the capsule. Uncapsulated bacteria has these antigens that are interacting with these receptors. It's been opsonized by these complements, and then these complements persist phagocytosis. Whereas this hasn't encapsulated, the complement can't bind with the antigens and the phagocytosis doesn't occur. These are some within phagocytic cells, some ways bacteria can exist. Some actually exist in the phagolysosome with the lactic enzymes in low pH environments. Some are just intravacuolar. They inhibit the maturation of the phagosome, so it doesn't bind with the lysosome and it just lives there. Others escape the phagosome into the cytosol. I think this diagram is too much. It's just showing you the different bacteria and how some are in the cytosol, some are intracellular, and some are both. I don't think you need to worry about that too much. There's something called intracellular motility. This is how the bacteria actually move from cell to cell. So it's an actin-mediated motility. that allows movement from cell to cell. It's not like the flagellum microtubules. It's actin mediated, and it steals host actin, which is part of our cytoskeleton. And on one pole, it has binding proteins, which facilitates polymerization, which allows it to move. And they avoid extracellular immune molecules because they stay intracellular. So this is the membrane between two cells, and it's just piercing that. Right, you can see the polymerization here is allowing it to invade this other cell where it's internalized in another vacuole, and the process reoccurs. What is intravacuolar? So intravacuolar is when a pathogen blocks phagosome maturation and as a result fails to fuse the lysosome, so the pathogen is not destroyed. The pathogens are protected. Some have bacterial secretions underpin this process, such as type 4 secretion system. This is like a molecular injection, which we'll talk about in a bit. And this is involved in TB, Legionella, and Salmonella. Others, this can be like latent for quite a long period of time, as in TB, and then when a host is immunocompromised or when the time's right, they can reactivate. Others can reside in mature phagolysosomes. They just resist killing. So these are just really hardy bacteria such as Staph aureus and Coxiella. These pathogens catalyze the breakdown of H2O, which is the reactive oxygen species, into water and oxygen. They're resistant to low pH and sometimes facilitate reactions that increase the pH, and they release proteases that break down antimicrobial peptides such as defensins, which try to punch holes in the bacteria or have antimicrobial effects in a variety of ways. So these are very hardy and not very high yield, but you should know that some can exist in phagolysosomes, some just exist in a phagosome. This is just showing you the life cycle of pneumophila. It's an intravascular secretion, so it goes in the phagosome. It releases effector proteins through this molecular injection, releasing proteins. The proteins then affect the cell cycle, which inhibits the maturation of the phagosome, so it doesn't combine with the lysosome and get degraded. It can then proliferate and then eventually invade. Scary stuff, not that important. I would almost say it didn't come up. There are different types of molecular syringes that are used for effector proteins to be released. They also have a variety of other mechanisms. They can affect DNA and DNA protein complexes. The reasons why these are important is they specifically punch holes in eukaryotic cells and put their toxins or effector proteins in a particular cell. So it's not wasted. It is very specific. I've said that they can release effector proteins to influence DNA. Sometimes they influence uptake, sometimes they release toxins. So they can release effector proteins which trick the cell into taking the pathogen inside. Some have needle tips that can punch holes to release protein inside, and it inhibits just secreting blindly specific. They have an example of Legionella. It's gram-negative, and it promotes uptake into the phagosome through the use of a type 4 secretion, which is this one. You don't really need to know what it looks like. And it just pumps a bunch of effector proteins, which will affect the cell cycle and maturation of the phagosome. So it slows maturation, lysosome fusion, and the acidification of the phagolysosome. We're almost done. Third last slide, I think. There's a zipper and trigger invasion mechanisms. These are the two prominent mechanisms of invasion in the human body. The zipper mechanism refers to a bacterial surface adhesion that interacts with a host cell membrane receptor to promote uptake by host cells. So often adhesin is a protein expressed by bacteria, and it binds to integrin or glycoprotein and tricks the cell into uptaking it. The membrane will curve and engulf, and that process looks like a zipper around a pathogen. I mean, it kind of looks like, it really doesn't look like it, but these people say it looks like it, so it looks like it. And examples is Listeria and Neisseria. And then there's an endosome, which is formed in the cell, which allows trafficking, proliferation, or dissemination. So the endosome is just what the bacteria exists in within that cell while it proliferates until it eventually disseminates. Maybe it does look like a trigger. I haven't actually seen it, but often they say things look like things, which they just don't. But maybe this is an exception. A trigger mechanism refers to the molecular syringe. We've talked about molecular syringe. This one is particularly TSS3. It injects effector molecules into the host cytosol and triggers enter by the formation of a vacuole. I have a picture of this, a picture of both of these in the next slide. It leads to actin polymerization in the cell membrane, which will eventually engulf the bacteria. These are the steps: molecular syringe pierces, pumps in effector proteins, encourages uptake, and it's specific, so it doesn't waste. This is a zipper. So I always take the pictures. I try to take the pictures from the lectures when, in these particular in micro, sometimes the examination questions are from the pictures, so I try to put them in. You have the adhesion proteins combining with receptors, the polymerization of the actin occurring. No, that's the wrong one, sorry. You have adhesion combining with the integrin and the glycoprotein. This triggers uptake and endosomal trafficking in this little kind of like empty cell, and that's either going to proliferate and then eventually disseminate. The one with the actin polymerization is this example. This is quite a complicated diagram, but you can imagine it's pumping proteins in. And imagine for a second that there is a little, imagine here, you have this molecular syringe, it's pumping in, and as a result, actin is polymerizing to change its shape around the pathogen to allow it to come in. We're almost done. Final slide is, if in doubt, choose Staph aureus, because Staph aureus has everything, essentially. These are all the virulence factors it has. It has a polysaccharide coat. It degrades IgG complement. Maybe it's worth, superantigen is important to remember about it. Essentially, all you need to remember is Staph aureus does almost everything. So that's like the final message. If you're in doubt, choose Staph aureus. The final slide today is, this is a clinical resource Peter recommended. It's from a Vespa. So Peter, so there is a club called Vespa, which do case nights. This isn't that. PSPs used to be called Vespers. So I think Peter found one from ages ago, and he recommends their clinical resources. So we just thought we'd share that. Thanks for staying with us, everyone. Are there any questions?
David, I might just jump in with a bit of an example for the pop health. I had a few questions, asking for some concrete examples. That's okay.
Yeah, of course.
So just like kind of the four main ones, I wrote up a little sample experiment. So let's say we're doing an experiment on the effect of exercise, which is our exposure, because that's the thing that they're exposed to, on depression. And that's the outcome. So that's like what they have. And I just listed down the four kind of main ones that people get confused about. So if that's my experiment, in a cross-sectional, I would just go out and I'd grab 100 random people, and I would calculate how many are depressed, how many exercise, and then see if there's some sort of relationship. Pretty much cross-sectional just shows you if there's a connection. It doesn't show you what causes what, because I've just grabbed 100 randos and saw if they're depressed or exercise, if there's a connection. In a case control, it's when I start with the outcome. So in this case, the outcome is people who have depression. So I go out and I find 100 people with depression. And then I see how many exercised, which would be our case, and how many didn't exercise, which would be our control. But I start with the outcome. With a cohort study, I start with the exposure. So instead of finding 100 people who have depression, I just find 100 healthy people. Specifically 50 who are about to exercise or who do exercise regularly, and 50 who do not. So that's my exposure. And then for a prospective study, I just see how many develop depression in the future, because prospective is all about following them, following them exercising, taking mental health samples, and seeing how many develop depression in the future. In a retrospective, same sort of thing. You start with exposure, but kind of the difference is, in prospective, you sort of usually start just before the exposure or like at the start of the exposure. Whereas in retrospective, you start once they've like been exposed for a bit, like for a while. So in retrospective, we would find 100 people, 50 who are like regular runners, regular exercisers, and 50 who are couch potatoes, like for a long time. And then from there, after I find my exposed group and my non-exposed group, then I also still see how many, based on that, have developed depression. But I'm still starting with the exposure for both prospective and retrospective. I can upload this on the slides as well. Just like give you an example, sometimes it's like a lot easier when you have like an actual experiment and then like you can see how it works. And just a reminder, case control is odds ratio, prospective and retrospective is relative risk, but we'll go through that in a future PSP.
Yeah. I'm going to be honest, it doesn't get explained much better than how Katja has explained it today. So
I don't know if that was a compliment or not, but thank you.
it was a compliment.
Okay, sweet.
Like, yeah. So I'd look at her resource. I know when we do this again, I'm going to look at her resources. So I'd recommend you do too. Thanks for coming, everyone. If you have any questions, chuck it in the chat. Otherwise, have a great day.
Enjoy your Sunday, guys. You and me, David.
Oh, I have to stop recording. How do I stop?